polypeptide is the linear string, and then it folds up into the shape of a protein.
So one is folded and the other is not?
That's a reasonable way of thinking about it, yeah. Question?
Can an amino acid take on only one form, whether the helix or the sheet?
No. So can an amino acid take on only an alpha helix or a beta sheet conformation? No. So some of the amino acids, like the notorious proline, really like to be in turns and are almost always found in turns, the periphery between the alpha helix and the beta strand. But basically, some of them have preferences. Like, for example, alanine is 2/3 of the time found in an alpha helix, but 1/3 of the time is not.
But a length of amino acid can, at any given time, can only have one.
Can a length of amino acid only have one conformation?
At one time.
At one
It doesn't
time.
change halfway through to another one.
So it depends on the chemistry. So there are stretches of 5 and 6 amino acids that are sometimes formed in alpha helix conformations and sometimes found in beta strand conformations. And there's very interesting biology that happens when an amino acid goes from being in not an alpha helix into an alpha helix. And in fact, that's how the flu virus gets into your lung epithelial cells. There are amino acids that go from being in a turn into an alpha helix, and that basically shoves a knife into the cell and allows the virus to get into the cell. So there's very interesting biology that underlies that chemistry. Okay, and I talked about how folding of the molecule makes up a binding site. And we did our little experiment in protein folding. So now we need to think about the flu virus. Okay, we saw this. This is our flu neuraminidase is the name of this enzyme. And this neuraminidase enzyme has these deep pockets. These pockets are called the active site of the enzyme. That's where this neuraminidase breaks bonds. Okay? And it does that in order to allow the flu virus to escape one cell and go and invade a neighboring cell or a neighboring child or something. So it's basically how the flu virus propagates its biological life cycle is it gets out of one cell, it gets into a cell, it makes copies of itself, and then it goes on to a different cell. Okay? So this is called a drug target. It's a protein that you can exploit for if you inhibit these red patches. If you can stick some chemical in the red patches in the flu neuraminidase, you can break the life cycle of the flu virus. Okay? And this has been done successfully using the techniques that we've talked about today, the protein chemistry questions, the X-ray crystallography questions. So this is the receptor on your lung epithelial cell for the flu. I mean, its job is not evolved to bind the flu virus, but you have these small molecules on the surface of your lung epithelial cells. They're called sialic acid ligands. And what happens is the flu neuraminidase binds to this shape. Okay? So how are we going to cure the flu? We are going to make compounds that look kind of like the ligand that the flu virus really likes to bind, except they're going to gum up the mechanism of the neuraminidase on the surface of the flu. Okay? And there's a couple compounds, Tamiflu, Relenza, that are basically small molecules, and what they do is they sit in that patch on the surface of the flu neuraminidase and gum it up so that the flu can't bind sialic acid, so the flu can't do its job that's required for propagation of the virus in its life cycle. Okay? And that ends up being a bunch of pharmaceutical chemists and medicinal chemists who are looking at the structure of the flu neuraminidase, the structure of the receptor for the flu neuraminidase, and making compounds that are mimics. Okay? And so I mentioned that different amino acids have different propensities to interact. So these histidines, these glutamates, they want to interact with their receptor. And so chemists at drug companies can exploit those interactions to make compounds that break the life cycle of the flu. Question?
Do the molecules just randomly bump into the flu virus, or are they somehow directed?
Right. So, so the sialic acid is on the surface of a cell. So it's not floating around. It's anchored on the surface of these cells that are in your lungs, and it's not going anywhere. But the flu virus actually takes the anchored sialic acid and liberates them. So there's some amount of this floating around. But no, when you take these compounds, yeah, you have to get them into your lungs. So one of the things the drug companies do is work on ways of delivering these compounds to where they need to go. So in the case of the flu virus, in the case of the common cold virus, all the action is in the lung epithelial cells. So you're looking at inhaled mechanisms for treating them and so forth. But if you're talking about cancer, if you're talking about some other disease, the delivery of the drug ends up being a different, you might use a different avenue for delivering. Question?
Do all the different kinds of flu have the same whatever,
Yeah.
red spots?
Right. So that's a terrific question. The question is, do all the different strains of flu have the same two proteins, hemagglutinin and neuraminidase, which are ridiculous names for proteins? But yes. So it turns out that right. So we've heard about H1N1 flu, H5N1 flu. So all of those differences in all of the strains of the flu, N in this case is neuraminidase, H is hemagglutinin, the ridiculous names of the ridiculous proteins. But basically, the red stuff does not vary. It is absolutely conserved because that's required for the neuraminidase to bind to a sialic acid and cleave it. But everything else is subject to change. And that's why you have to get a flu vaccine every year, because you might have a vaccine that allows you to recognize this particular set of amino acids, but then the flu virus recombines. There are these mechanisms for evasion of the immune system that the flu viruses have. They're called antigenic drift and antigenic shift. And every 11 years, it turns out there's some recombination event where the flu presents a very different surface, and that tends to be associated with very severe flu seasons. But basically, to answer your question, this part never changes because that stuff is absolutely conserved to do the job that's required for the life cycle of the flu. The rest of the stuff is just a decoy for the immune system, and that's why we have to get vaccines every year. Is there a question back there still?
Yes, you have these protein molecules in the on the surface of the lung that they bind to flu virus binding to, correct?
So it turns out the sialic acid, what flu virus binds to is not a protein but a carbohydrate. But there's a receptor on the surface.
But those carbohydrates are there for a reason other than to bind to the flu.
Yes.
And this drug that you're putting into attack the flu, does that come up or change how those things bind to what they need to bind?
Yeah, I mean, that's a terrific question. So the question is, if you're ingesting analogs of sialic acid, isn't that interfering with the normal function of sialic acid? Most of the time, sialic acid is found on the surface of cells, attached to proteins, attached to lipids. Most of the time, it's not floating around. And it turns out it's a terrific question, and that's why you don't take Relenza all the time. You take it in order to treat a disease. But sure, anytime you take a molecule that looks a lot, you ingest a molecule that looks a lot like something that is important for the biology, you're introducing the possibility of side effects. It's absolutely true.
Yeah, obvious question is, why can't you just use sialic acid and just overload it and you just clog up viruses before they can even get into cells?
Right.
It's a natural substance that's in my lungs.
Right. So the question is, why can't you use sialic acid as a drug for treating the flu? Right? We know that the neuraminidase on the flu bind sialic acid, but it turns out these guys are actually better binders. So the flu has evolved not just to bind to these receptors, but to let go at some point. And so it's actually easier to get tighter binding in these sialic acid analogs, and tighter binding means you can bind and kill the flu in a way that the sialic acid is basically called a low affinity or a weak ligand, and the drug compounds are tighter binders to those surface proteins. Okay, so we've talked a bit about flu, but of course the common cold, we haven't cured that. So drug companies, pharmaceutical companies have developed treatments for the flu. You can get vaccines, you can get these sialic acid analogs that work to gum up the hemagglutinin and the neuraminidase. So now here's the rhinovirus, which causes the common cold. So in this case, it's a very beautiful icosahedral virus. It looks like a soccer ball or whatever, but there's no hemagglutinin, there's no neuraminidase, the receptor is not sialic acid. So the same principles are in place. The virus binds to a surface marker, a surface receptor on the lung epithelial cells, gets into the cells, takes over the cells, makes copies of itself, and then goes elsewhere. But we're not as far along with the common cold as we are with the flu. So we have the same principles in place. We can look at this surface and look at the proteins on the surface of the rhinovirus and say, look, oh, there's a nice channel, there's a nice channel. Right? So we're following, we're looking for cures for the common cold using these principles that we've talked about, but we're not there yet. And that's why the question that we're tossing out is, can we cure the common cold using these principles? Okay, I think I'm out of time. I will stop there and take any questions that anybody might have. Question here.
I see there are star shapes in the blue patches on there. Are
Yes.
those all identical? Are they the same or are they different?
Yeah, so you must be a soccer player or something. So the question is, are the blue patches all identical? Yeah, so viruses carry around a relatively small genome, and one of the things they have to do is encapsulate their nucleic acid. And so one efficient way to do it is to use a relatively small number of proteins, but have lots of copies of that protein. And it turns out these viruses are called icosahedral viruses. And an icosahedron has a 5-fold axis and a 3-fold axis and a 2-fold axis. And so does a soccer ball. And basically, nature has found a way of making not carrying around a lot of genetic information, but using lots of copies of the protein that comes off that genetic information in order to make this package, in order to deliver the nucleic acid to the cells, which allows that virus to propagate. So yeah, it's a principle of conservation that a small amount of genetic material can lead to a nice icosahedral package and protect the nucleic acid. Yes, the common cold is caused by this thing, which is called the human rhinovirus. And it is a virus, that's right. Do any of the proteins act like a flu virus? So, yeah, that's an interesting question. So it turns out some vaccines are actually attenuated viruses. So if you have a vaccine, it's got protein in it, but no nucleic acid. So the way these viruses work is they have proteins that protect the genetic information, and if you have just the protein part of a virus, it can work as a vaccine. And so it won't propagate to other cells, but it will certainly interact with the chemistry on your cells. Yeah. Right. So the question is, it seems like it's hard to get proteins to crystallize in order to get these atomic images, and that is absolutely true. And there's some people in my lab who would be happy to commiserate about how hard it is to crystallize proteins. But yeah, so growing a crystal of a salt is easy, right? But growing a crystal of a protein ranges from being difficult to impossible, depending on what protein you're talking about. And so what we do is, anybody know how to grow rock candy? Who's grown rock candy? How do you grow rock candy? Your parents made it. Was there heat involved? Yeah. Right, you take about this much sugar, you pour it into boiling water, you saturate that solution with whatever you're trying to crystallize. And in the case of sugar, you can use heat. And so you basically heat it up, you slowly cool it down, and then you drop a string in there, or you basically nucleate it, and then you get crystals. So we're using these same techniques of you make a supersaturated solution, you slowly, if you make a supersaturated solution of a protein and slowly drive the concentration up, if you're lucky, you can get the protein to crystallize. We don't use heat because heat will actually denature proteins, right? When we cook eggs, when we cook egg whites, if you add heat to proteins, it often makes them go from being folded back into the linear form, and that's not what, that'll never crystallize. Okay, so I did bring a tray. So this is how we do our protein crystallization experiments, and we're basically working at the cubic millimeter scale or less, and we're basically slowly saturating a solution with protein, and if we get that right, we can grow crystals. And but it fails 99% of the time at least. It is hard.
I didn't get the answer. Can we expect to cure the common cold?
Yes, I'm an optimist. I would say these techniques will get us to treating the common cold. Curing, that's a harder thing. Certainly, we will get better treatments for it. Chicken soup. Chicken soup contains sialic acid, as it turns out.
I remember hearing a long time ago that there's like some finite number of versions of the common cold, 130 or 140, and when you get a cold, from then on you're immune to that type. So if you could ever live long enough to get 140 versions of the common cold, is any of that true, or is that all just
You're talking about rhinovirus or flu?
Yeah, common cold.
Gosh, I don't know how many versions of rhinovirus there are. So that it depends a little bit on does one amino acid change? Is that a different strain or is that the same strain? It turns out to your immune system, changing one surface amino acid can change the personality of that protein and can make an immunogenic protein non-immunogenic or vice versa. So my guess is living long enough won't guarantee to protect you against every common cold. But that's a good experiment. If you live to 130, you can do that test. Yeah.
I'm wondering how you get from the X-ray picture of a cross to the bizarro helix, or whichever direction it goes. How do you get from a cross to a helix?
So that is a Fourier transform. It's basically there is there's mathematics that gets you from one space into the other, and I guess Fourier transforms we see in other instances. So